Welcome back, class. In today's lecture, we're going to explore an important part of the field of microbial ecology. And we're going to do this by discussing the process of elemental cycling. That is the exchange of nutrients by microorganisms that makes life on Earth possible. This is sometimes referred to as environmental microbiology. We're also going to look at some of the more important tools and techniques that are used to study microbial ecology. Let's now consider the physical environment, because as you can imagine, microorganisms, as they interact with each other and with other organisms in biogeochemical cycling, they're also influenced by their immediate physical environment, whether it's soil, water, deep marine environments, or even a plant or an animal host. So it's important to consider the specific environments where microorganisms interact with each other and interact with other organisms and the physical environment. Let's turn our attention now to these terms microenvironment and niche. So when we're talking about the specific physical location of a microorganism, this is called the microenvironment. And in this microenvironment, the flux of required electron donors, electron acceptors, and nutrients to the actual location of the microorganism can be limited. At the same time, waste products might not be able to diffuse away from the microorganism at rates that are sufficient to avoid growth inhibition by high concentrations of these waste products. These fluxes and gradients create a unique niche, which includes the microorganism, its physical habitat, the time of resource use, and the resources available for microbial growth and function. In this illustration, we can see the creation of a niche from a microenvironment. So we have two nearby particles shown in brown, and these create a physical microenvironment for possible use by microorganisms. Chemical gradients, as with oxygen from the oxic region, shown in blue, and sulfide from the anoxic region, shown in pink, create a unique niche. And this niche is the physical environment and the resources available for use by specialised aerobic sulfide oxidizing bacteria in this particular example. Of course, microorganisms contribute greatly to the functioning of ecosystems, and you might remember that ecosystems can be defined as communities of organisms and their physical and chemical environments. And these communities function as self-regulating units. So these biological units respond to environmental changes by modifying both structure and function. Now, microorganisms in ecosystems can have two complementary roles, as listed here. One, the synthesis of new organic matter from carbon dioxide and other inorganic compounds during primary production. And two, decomposition of accumulated organic matter. The general relationship between primary producers that synthesize organic matter and decomposers was once thought to be quite simple. It was held that different organisms performed these non-overlapping processes. But we now understand that microbial communities are more complicated than that. And because aquatic systems are easier to investigate than terrestrial microbial communities, much of our current understanding of the contributions of microbes to ecosystem function comes from studies of these environments. So you can imagine that larger plants and animals contribute to a common pool of dissolved organic matter, DOM. And this dissolved organic matter is consumed by a variety of prokaryotic and eukaryotic microbes. These microbes then return some of this dissolved organic matter to larger animals in the form of particulate material. That is to say, protists eat bacteria and primary consumers eat protists. In addition to that, the metabolism and death of these microbes recycles some of the organic matter back to the general pool of dissolved organic matter. So this complex web of interactions is referred to as the microbial loop. Scientists known as microbial ecologists study natural microbial communities that exist in soils, waters, or in association with other organisms. But regardless of the habitat they study, these scientists seek to answer several fundamental questions. First, they're interested in the microbial population. How many microbes are present? What genera and species are represented in the ecosystem? Then once these questions have been addressed, community dynamics can be explored. For example, how do these microorganisms interact with one another, with higher eukaryotes, and with the abiotic features found in the environment? The nature of microbial ecology has generated an enormous amount of interest from various disciplines, and as a result it's also generated an enormous assortment of methods, including microscopy, physical techniques, chemical techniques, and in particular now molecular techniques. So in the following slides you'll find an overview of methods used in microbial ecology. Note this material will not be assessed in the test, and there is no audio associated with these slides. However, for your interest, I've included some additional notes where required in the notes panel at the bottom of each slide.